thank you very much, Imogen. It's absolutely great to be here, and it's the first conference I've been to in person since all the COVID stuff, so it's fantastic to see everybody. So when I was asked, what is a clinical trial, everything we should know, I thought, oh, that'll be easy. I've been doing clinical trials for ages, know lots about them, I've done various talks on them. And then I thought of the audience. Now I'm frightened. I don't know what you should know. So I thought to myself, how shall I structure this? I kind of went through the questions people have asked me in the years gone by. What are the things people ask? What do the research nurses get asked? And was going to go through a list of frequently asked questions. That then became quite a jumble. So what I'm going to do, I'm going to try and structure it in what's a clinical trial, try and describe that, talk through the history, because I think it's quite interesting, and then talk briefly about what are the different types of clinical trials, the different phases of clinical trials, talk a bit about a journey of a trial, so how does a trial get set up, what happens across the whole trial, and then I'll go through some of the sort of more specific questions like, should I take part? How do I find a clinical trial? What happens if I'm there? And then talk a bit about the, if I get time at the end, talk about the networks and some of the regulatory stuff. Okay, so what's a clinical trial? So the WHO definition, sorry, I was trying to see my slides, I've got them there. The WHO definition is a scientific way to try and work out whether an intervention helps a health outcome. So that intervention could be anything from drug treatment to a test to an operation, exercise, any of those sorts of things. And then the outcome could be kind of survival, prevention, quality of life, side effects. So, you know, a trial could be, does this treatment make you live longer? Does it cure more people? Or a diagnostic test might be, if we use this test, can we detect cancer earlier, and will that make a difference to the outcome? And that outcome could be quality of life, could be survival, or other things. Does an operation give a better cosmetic output at the end? So all of those are interventions, and those are trials. They're different to observational studies, but observational studies are also important. So so a lot of research is observational studies. You're not actually doing something to somebody, but you're studying things as they go along. And you know, one of the classic ones was Richard Doll's working out that smoking caused cancer. So what they did in that was looked at everyone who'd got lung cancer, studied them, asked them what they were doing and not doing, and then worked out actually that it's probably associated with smoking. Then they did another really long study where they took a load of doctors and followed them up over many years, looked at their behaviors, and then worked out quite easily that smoking is what caused cancer. And there's a lot of that for melanoma. So a lot of what we know about melanoma is based on observational trials, so observational studies. So the business about, does sunlight cause melanoma in some people? That was done by observational studies, looking at sun exposure and trying to work out which people, so taking a load of people who'd got melanoma, doing questionnaires and following them over time. And you realize that actually, with patients, people who have melanoma and people who don't, sun exposure is a key factor, at least in some people. And other observational studies were things like trying to work out when that sun exposure, so they looked at people when they emigrated to Australia and worked out that actually the sun exposure when you're young is probably more relevant than otherwise. So a lot of what we know is through observational studies rather than interventional ones. An intervention in that setting would be doing a questionnaire, not doing a questionnaire, doing a campaign to stop sun exposure. That could be an intervention, and that you might measure less people getting melanoma. One of the current ones going through at the moment is, does the microbiome affect outcomes following immunotherapy? So there's quite a lot of data now from the laboratory which suggests that what's going on in your gut affects the immune system, and that can affect things. So there's observational studies where people who are going through immunotherapy are being asked, can we study you? You do a questionnaire of your diet and lots of other things, but also within that, can we have some of your poo? And that gets looked at and analysed. And then in years to come, we'll be able to go, oh, actually, that sort of poo, or bugs in that sort of poo, people did better than others. So there are observational studies as well, which aren't quite clinical trials, but I'm going to focus today on clinical trials. So the history of clinical trials is interesting, and lots of people have kind of looked at that. So I guess people have probably been experimenting for years and years, but the first recorded stuff is in 500 BC in the Book of Daniel. And this is where the Israelites at the time were being put into, there was a siege or something, and they were being given food which had Philistine stuff in it. And Daniel went, hang on a minute, we can survive, we don't need to eat this, and did an experiment where he took people, gave them a vegetarian diet for, I think it was 10, 20 days, and then, or the meat-full diet, and people realized that you could survive not having to have all of the food and the meat. So that's described as the first clinical trial. I'm not sure if that's actually right. And then in Persia, Avencia, who wrote a very large tome on medicine and lots about medicine, and within that there is references to how do you test new medicines. So the stuff around clinical trials has been, I'm sure, going on for quite a long time. The person who's credited with doing the first randomized, well, it's not a randomized trial, the first trial was James Lind. He was a naval officer in the 18th century, and during that time, one of the really big problems was in the Navy was people getting scurvy. So scurvy is a condition you get if you don't have vitamin C, and it's a fatal condition. You get lots of bleeding, your resistance to infection goes down, and people had been trying to work out what causes it and how could you prevent it. And they thought that you needed acid in your stomach to try and do that. So what he did was took 12 sailors, well, they were out at sea, and they'd been out for a while, and as people started getting scurvy, he took 12 people who had scurvy and gave them 2 each of, I think it was cider, I'm just trying to read my writing here, vinegar, hang on, can I read all that? But some of it was citrus, orange and lemon. And actually looked, 2 of them, the people who had the vitamin C, the oranges and lemons, actually survived and could go back to work. The others, unfortunately, succumbed. So that was a trial, very positive trial. Actually, it goes to show you don't need hundreds and hundreds of people if you've got a really strong effect. And he wrote that up. And interestingly, though, although that's written up as a great trial and things, it took a very long time before it became accepted that that's what you did. But it changed the Navy. I think more people were dying in the Navy, in the Royal Navy, from scurvy than they were from being killed by the Spanish and the French at the time. So that was the first trial. Around that sort of time, there was also another study, and the thought of placebo came in. So John Haygarth was a surgeon and around that time, there was a doctor who was thought to be a bit of a quack anyway, called Leicester Perkins, and he developed these metallic, one steel and one brass, and they're called tractors, basically. They're pincers of sorts. And you could use them for most things. You could lance boils with it, but if you rub them over joints, you could make joints better. And he seemed to get a lot of people believing that. And John Haygarth thought, well, hang on a minute, this is complete nonsense. Let's show and prove that this isn't right. And he took wooden tractors, painted them brass and silver, and did an experiment on people with rheumatism. And he took 5 patients. He first treated them with the placebo, and then with the other, and they measured it. Somebody else measured whether their joints got better, and he showed for the first time that a placebo effect was there. And actually, they stopped using the Perkins tractors. And then the first of the modern trials was in the 40s, and that was when a new antibiotic had come in, streptomycin, as a treatment for tuberculosis. And the Medical Research Council set up the first randomized trial where they took patients, gave them either a placebo or streptomycin. They didn't know at the time whether streptomycin would work, and treated patients in a very, very systematic way. And it's based on that pedigree, if you like, that the Medical Research Council and then all the other things have set up trials looking at things. So that's the history of kind of what's brought us to where we are. So you can think of trials in different ways. So we're going to talk about the different phases of trials, but I'm also going to talk about the type of trial. Are they single arm, multi-arm? You hear lots of different phrases when you talk about trials. Are they controlled? Are they placebo-controlled? What's blinded? What's not blinded? What does all that mean? So I'll start with the kind of type of trial. So trials can be single-arm. That means all the people get the treatment, or get the same treatment, and you're studying them as you go along. Or they can be multi-arm, where you're comparing. Oops. Ooh, hang on, just trying to. Yeah. Okay, where you're comparing one arm with the other. Usually it's 2 arms, but it can be 3 or 4 arms. When we say arms in trials, we mean groups of patients getting allocated one treatment or another treatment. So having a conversation with a patient where I was explaining trials, and very, very intelligent guy who seemed to be getting everything I was saying. And then I told him about he might either get an infusion. And then I started talking about arms of trials. And he looked at me blankly. He was trying to understand what I was saying. And he was thinking, hang on, I'm getting one arm, I might get this treatment. And then in the other arm, you're going to get 2 treatments. And he was trying to work out what I was trying to say. So when we're talking about arms, we're talking about groups of patients getting a particular thing. And they can be randomized or... so when you're trying to allocate what a patient gets, so if you're comparing 2 different treatments, to take away any bias, they're allocated a treatment at random. And that means patient doesn't choose, we as the physicians don't choose, and actually it is done randomly. and that's generated by the computer. So sometimes people have the sense that, the computer is, you're putting in all of my details and the computer's choosing the best treatment for me. That's not what's happening. You are literally randomly being allocated a treatment or not, and that's to take away any bias. And that's a mechanism which is there to take away that bias and works very well to try and do that. Then the other thing is some trials are blinded, meaning you know that you don't know which treatment you're getting. So you might have infusion, you might have drug A or B, and you don't know which one you're getting. So you're blinded. Often if the treatment's blinded, not only are you blinded, but the physician's also blinded. So you don't know, and we don't know which treatment you're getting. You're followed up exactly the same. We know how to treat the side effects, whatever you have. And that, again, is to take away bias. And there is a real bias if you know which treatment, or potential real bias if you know which treatment you've got. So if you have the established treatment or the new one, which you think's better, you might want to keep on going. As a patient, you might say, this is a better treatment. I'm going to ignore the side effects a bit more, or just a bit more tolerant of it. Or if you're just getting the standard treatment, you're going, this isn't working for me. So there's an element of how much you report. As a physician as well, actually, it can add a little bit of bias in how you're interpreting either side effects or whether you do scans a bit earlier or not. So it does take away bias. And in an ideal situation, you would make it double-blinded if you can. And then controlled, when they say controlled, it means you have one group of people who are getting the standard arm of what your normal treatment is. That's the control group. They're the ones who you're comparing it with. And that control group is often the established treatment. So, if you were looking at immunotherapy, you might use the standard immunotherapy, and then that's the control group, and then the other group get the experimental therapy. The control group might be a placebo. So if there isn't a standard treatment at that stage of the melanoma, you might do a placebo. Again, that's to take away the bias of having a treatment. There is a placebo effect. Ooh. I'm not sure I'm not sure which slide set I've raised. But I'll go with this one. Sorry, I updated my slides as well, and I've got 2 or 3 versions. Okay, so then I'll talk a bit about phases of clinical trials. So it's worth thinking about that is how is a treatment get into being an established treatment? So it spends a bit of time in discovery, during the discovery phase, and that's in the laboratory when a drug's developed, initially completely in the laboratory, doing often with cells or cell lines, and then when that looks, so a way you might do that is taking a new treatment or a new drug, looking at that on melanoma cell lines, which you can grow in the laboratory. Does it kill them? Does it not kill them? And then trying to make the drug as good as it can be. Then you'll test in animals. Does it work? Is it givable? Is it safe? You'll then work out the dose and how the body deals with it. And then you'll go into phase 1 usually. There is something called a phase 0, but the main trials are phase 1, 2, and 3. And the phase 1 trial is when it's usually a first in man, and I'll talk about that. You're trying to see, is it even safe to give, and can we give it to people? You're then looking, once you've shown that it's safe. You're then going into a phase 2, trying to see does it work and does it work enough. And then finally, you're looking in a phase 3 study to see is it better than current treatments. How long that takes varies quite a lot. Usually a phase 1 trial might take a year or 2 years. A phase 2 trial, probably 2-3 years all in. And then a phase 3 trial can take 5, maybe even more years, because you're looking at results which are a bit longer. But I'll talk about each of those phases. So I'll talk a bit about the different phases, and I'll start with the phase 1. So a phase 1 trial is when you're looking to see, so you've tested a drug, you've seen that it seems to work in the laboratory. That's probably taken about 3, 4, 5, 6 years, something like that, developing it into a compound that you can give initially to an animal, and you'll test it in a mouse. So there are mouse models, they call them, but what they are is mice who have and develop melanoma, and you can test, does this kill the cancer and still allow the mouse to be alive. And while you're doing that, you increase the doses in the mice to see what sort of a dose you can get. You're then in a stage where you're going to take it into humans. So, is that it? Fantastic. And you found where it is. Thank you. Brilliant. There you go. Okay, sorry. Thanks. And it can be a first in man, or it can be the first time it's been used in a particular way. So we're saying first in schedule, or it can be first combination. So the first in man is the first time you're using it. And the goal there is to try and see, is it safe? Can you give it? And to get the optimal dose. So actually in that phase 1 trial, you're looking to see, is it safe? Can you give it? And hoping, but that's a secondary output, to see how well it works. Small number of patients, 20 to 50 usually. Sometimes it specifies which cancer it is, so it'll be a melanoma specific, but often it isn't, because some of these drugs work across different boundaries. So they might work in different cancers, so you can test it in almost all comers. It's usually when there's no standard treatments left for people. They still need to be well enough and fit enough to be able to get them, and it's usually done in specialist centres. If you want to know anything more about phase 1 trials, actually Sarah Danson is the national lead on phase 1 trials, so questions to her later on. So again, what you're trying to do is find out, is it safe, what's the right dose, and what are the side effects? So what you do to find out what the right dose is a thing called dose escalation. So you start off at a particular dose, and that's a tenth of the dose which you just started in mice or which were lethal in mice. And there's a way of working out how do you convert from animals into humans. And you start at a particular dose. You start it in one patient, you wait for a period of time, and then provided that person's okay, you use the next one, and then you do the next one. If they're all okay, you go on to the next dose level, and if they're okay, you increase it again. And these are set as to how you increase the doses. And then if at a stage you get somebody, and you set out, there are ways of measuring the extent of side effects. So very, very standardized. You get grades of side effects. So a rash can be grade 1, 2, 3, 4. Sickness is measured in 1, 2, 3, 4. Your blood count being affected. There are grades of toxicity, and they're very well recognized, so that you can compare between one and the other. It's a scientific way of doing it. That's why if you're in a trial, you'll get your nurse asking you about the details of the side effect. You're going, look, I just feel sick. Yeah, but how sick? You know, and there's a reason for them wanting to do that. So if they get a dose-limiting toxicity, if one patient gets that, you then will carry on at that dose and see with another 3. We call it a cohort, so an expansion cohort. And if that then is those people are okay, you'll keep increasing the dose. But if you get 2/3 of the patients getting the side effects, you know that you've reached what we'd call a maximum tolerated dose. And you come down, and that's the dose you'd then use for your further studies. So that's working out what's your maximum tolerated dose. That's a phase 1 study, and during it, there'll be lots of close monitoring, lots of blood tests and hospital visits, so you kind of need to be prepared for that. And what we're trying to do is learn how the body's handling the drug, what the side effects are. You might get unexpected side effects, and you might have to wait for a slot. And you know, so there are some disadvantages. You might end up being in the low dose cohort. So there are potential reasons you might think, oh, do I want to go that? But actually, it's access to a drug which is not yet available. And certainly with a lot of the new melanoma drugs, when the ones we're currently using, there are lots of people for whom there was not a treatment, and the treatments that came in were absolute game changers for them. So, okay, so that's a phase 1 trial. So drug gets through phase 1, you're then looking to see what do you do. And the next phase is trying to work out, is there an anti-cancer effect, and getting a clearer idea of what the side effects are for that. And the endpoints, when we say an endpoint, is what you're measuring to find out at the end of the trial, is response rates, time to progression, and overall survival. Those are terms we use quite a lot, and I'll explain them as we go along. And the goal really is to see, does it work enough, and is it a good enough drug to then do a phase 3 study to see that it's to be established as a treatment. And this is usually about 50 to 200 patients in a phase 2 trial. And a phase 2 trial, often single arm or 2 arms to that study, may or may not be blinded, and often is specific for the cancer type. So it's usually there'll be a phase 2 study in melanoma. And what you're asking is there an anti-cancer effect? And we measure that in different ways. So one way of that is the cancer smaller? So, and in what proportion of patients? We call that a response rate. So if you hear news, oh, the response rate was whatever, it's measuring whether the tumor was smaller. And we have a very, very exact way of defining what we mean by smaller, actually. So what we do is usually we'll measure response looking at CT scans. So people will have a scan at the beginning, and then as we're going along in the trial. And this is a scan of a chest. Somebody's lying with their feet towards us, on their backs, and we've got a slice through the middle. And that's before and that's after. And the black bits are the lungs. That's the air in the lungs. That's the top of the chest, and that lump there is the metastasis. That's the melanoma secondary. What you do is you measure with a ruler across it, and you go, that's what the size was. and then you measure it afterwards. You measure up to 5 of those and add it up. If it's smaller, then you can then work out what's happening. So if it's bigger, we call that progression. I'm not sure progression's the right word. Progression sounds like a good thing, right? In any other bit of your life, if you're progressing, you're going forward. But we kind of ended up using the term progression for meaning cancer's getting worse. But it has to be about 20% worse to be progression, or it can be stable, or you can get a partial response where it's shrunk, but we have a cutoff when we say if it's shrunk by 30%, that's a partial response, or a complete response, meaning we can't see it at all. That doesn't mean it's gone completely because we can't see microscopic deposits. So you'll hear reports of, the response rate. So often when doctors are talking to you, they're talking about, the response rate is, and what they mean is that, for example, in 55% of patients, the tumor has shrunk by that amount. It's shrunk by 55%, but it's shrunk, right, by 30%. So you've got a partial response or a complete response. And in a trial, that might be, the standard treatment response rate was 25%, and it's gone up to 55%, so that's a good thing. We use the same thing, we call something called progression-free survival, and that's kind of a measure of how long is the cancer controlled for. So what it means is the time it takes from the beginning of your trial or beginning of your treatment to the cancer getting big enough to call a progression. So and that's measured in months, and we look at them and often it's reported as the average and the median average. With the progression-free survival might have gone from 18 months to 24 months. And then the other measure of how good a drug is overall survival. How long do people live if they had one treatment or another? So that's how we measure, is there an anti-cancer effect? There are a load of other things which are measured, which are about side effects. So you'd be asked about side effects regularly. They have blood tests to see if the drug's affecting your kidneys, your heart, your liver. You might have heart monitoring with an ECG. And these are graded and recorded. So that's what we'll do in a phase 2 study. We'll hopefully, by the end of it, know, is this drug, does it work? And does it work enough to make it worthwhile doing the next trial, which is a much, much bigger undertaking, a phase 3 trial. That can depend on lots of different factors. So there are quite a lot of drugs which get to phase 2, and they're not that much better than the standard treatment, or the side effects are much worse, so they don't progress onto a phase 3. So that's your first couple of phases. The next bit is what you're trying to do is to compare the drug or the combination of drugs with what's the best current therapy. And usually you're looking, the primary endpoint is, do people live longer or not if they have this treatment or the other treatment? Or does their cancer come back or not? And the sample size depends how much of an effect there is, but it's usually between 500 and 3,000 patients, that kind of size of trials. So those are usually multi-center trials done in lots of institutions, usually international for a cancer such as melanoma. For a cancer that's much more common, you might be able to get away with trying to do a phase 3 study in one country. And we have done. We're doing phase 3 studies in the UK. We've done some large ones, and so it is feasible, but often there'll be multi-center and international, and they'll always be randomized. Right? May or may not be blinded. So I'm going to talk about phase 3 trials. Ooh, bloody hell. Sorry. I'm talking a lot. We might go back to the old slide set. Sorry about that. Okay. So we're going to talk about a particular trial to illustrate what a phase 3 trial is. So this is Combi-V. So BRAF is a mutation in melanoma which we can target with a treatment called vemurafenib. And actually, sorry, just digress a minute. Somebody was very disappointed that they had a BRAF mutation because just the word, I've got a mutation in my cancer, sounds bad. You don't want to have a mutation, but actually all cancers have mutations, and they all have lots of mutations. So the fact that your letter says BRAF mutation isn't, I think, anything particularly worse or not. In fact, you have a different treatment option, but it's just, all cancers have lots of mutations. Sorry. So the standard treatment at the time was a tablet called vemurafenib, a real game changer. It had lots of side effects, but a new combination of dabrafenib and trametinib had been shown in the phase 1 and 2 trials to be safe and givable, and seem to be probably work a bit better. So the trial was set up, international study, comparing what it had was 700 patients getting vemurafenib to dabrafenib, and they worked out that they'd need about 700 patients because from the phase 2 they knew just how much of a difference there was going to be, and they sat down with statisticians who worked out, well, if you do a trial of 700 patients, you'll probably see a difference that's significant. Right? If you do too few, you just won't see enough difference, and you weren't sure if the difference was by chance, and you don't want to do too many. And the primary endpoint was overall survival, but there was loads of other things where people were looking at: response rate, safety, quality of life. So between June 2012, God, was it that long ago? 10 years ago now, yeah, and 2013, about 1,600 patients were screened, and about half the patients with melanoma have a mutation. So this is patients who had metastatic melanoma that had spread, couldn't cut it out, and they were entered into trial. The trial had 193 centres across the world. There were about 10 in the UK. So what happened during that time, patients were seen and screened and consented, and if they were eligible, they went into the study. Data was then continuously being collected. Safety was being closely monitored, and the study wasn't a blinded study, but they don't do the analysis. So some people think, oh, is the trial working? Is it showing working? We do monitor how the toxicities, so the side effects, continuously, but the people running the trial don't get to see whether it's working or not as you're going along. You don't do that data analysis because that might influence how you then behave. And so if you look at trials as you're going along, you sometimes will see a blip where one treatment looks a bit better than the other. And if you look too early, you get the wrong idea and can influence how good the trial is. So, but that is looked at by the Data Monitoring Committee. And they have set levels. So if the treatment is working really well, you don't want to continue giving the treatment because actually you want people not to be having the standard treatment, you want them to be having the other treatment. So the Data Monitoring Committee actually, as the trial went on, looked at it and said, look, this effect is really good, we should stop doing the trial. People continuing on the treatment, but they were also allowed to cross over and so those people who just had the vemurafenib crossed over to the other. And what the trial showed, and this is what data looks like when it comes out, this is the overall survival. And it's a graph. The blue one is, so what that graph is when you first start off, 100% of patients are all alive. And then as you go along, people die and that graph goes down. And you can see that towards the end, in the vemurafenib arm, about 50% of patients had died. But in the combination arm, more people were alive. So what that trial showed is that the combination works better. In fact, it gave us a lot of safety data and showed us it was a bit safer and showed us the quality of life was also better. It gave us lots of other information, but importantly, it gave us the data to get the drug licensed. Okay, so that's phase 3 trials. And basically, you're looking, does it work better, and what are the side effects? So I'm just going to go through how does a trial get designed, something like the vemurafenib trial. Basically, people sit down at meetings, and there are groups of people who are kind of spend their lives, Sarah's one of those, thinking about new trials. What are the questions we need to ask? What are the treatments which need to be tested? They design them, and now in that combination of people is statisticians, it's increasingly, it'll be doctors, scientists, and increasingly patients are involved really early on in that design. They then need to secure funding, go through a regulatory approval. All of this is nightmare. But there are people who are really good at doing that. Then you'll go to different sites to see where can these trials be opened. Trials are very expensive to run, so you can't open and regulate, so you can't necessarily open them in everywhere. Some trials you can, some you can't. You then need to recruit patients. Once the trial's open, you can start recruiting patients, collecting data. Then there's a period of time where there's data analysis. You're trying to work that out. And then finally, you look at the results, you publish them, and then there's a peer review during that publication of, is that the right thing? All of that is very tightly regulated, you'll be glad to know, and works, actually, works well. So, I thought I'd be done by about half an hour. So, first thing is, how do you find so I'll stop there about clinical trials and I'll just do some of the questions. How do you find a trial? And I know the team at Melanoma Focus are going to talk a bit about that afterwards. So the first thing is, I think, just ask your oncologist, and they may know, or they may know how to find them. I think that would be a good place to start. But there's some very good web-based trial finders. Clinicaltrials.gov, that's the US site, which keeps a monitor of all of the trials. It's a bit cumbersome, and it's quite hard to find the trial you want in the UK and where you'd want it. Cancer Research UK, if you look up Clinical Trials Cancer Research UK, also has quite a good website, and a particularly good one is the NHS one, which actually is working quite well, called Be Part of Research. Basically, you fill in what type of cancer, and they'll come up with trials and does it by postcode. But the Melanoma Focus one's now got a very good trial finder, which is a melanoma-specific thing. And I think, Susanna, you're going to talk a bit in a while about that, so I won't talk anymore. What will happen to me if I have a clinical trial? So the first bit of that is the doctor will sit down with you, with the research nurse, and go through the trial. Usually, or at least the way I do it, is I'll tell people what the standard treatment we would be using or what I'd be recommending, and that there is a trial which you might want to consider. It's completely voluntary, and then I try and go through what that trial is, what the advantages and disadvantages are as I see them, and then give them an information sheet with it. Information sheets are about, I don't know, 8 to 9 pages. We try and write them so that they're understandable and you can follow them. But they're hard, actually. They are hard. And if you end up with an information sheet and either you have difficulty reading or what you're reading isn't making sense, sit down with a research nurse and go through it with them. There's absolutely no harm in looking at it. And they're really hard. So don't be worried about asking. You'll then be asked to sign a consent form. Again, that has loads and loads of bits on it. I've read the information sheet. I'm agreed to have my data sent away. I agree to have, and it feels cumbersome. You just need to do it if you agree with it all. Then you'll go through some screening tests, scans, blood tests, make sure you're eligible. If you are, you'll then be allocated a treatment. That's called randomization. Sounds like something in Star Trek, doesn't it? I'm going to be randomized. That's basically being allocated a treatment. You'll then go through treatment, continuous monitoring, data collection, then you go through a cycle of that. And the treatments might be for a short time, might be longer term, but when the treatment's stopped, there's usually still a follow-up period where you'll continue being on follow-up. And then increasingly, people are given the feedback of the results of the trials. And you should ask that, actually. Will I get to know the results of the trials? So finally, should I take part? They're not right for everybody, and not every trial's right for everybody. So I think that the things to consider as the reasons you might want to take part: access to a new treatment. We've all of us as physicians have got patients who have clearly benefited from being in trials, and much more benefit than if they hadn't been in trials. And potentially better outcomes. So there's some interesting data to suggest that people in trials, even if they're in the standard arm of a trial, do better than people having the standard treatment not in a trial. And that's probably just about being monitored a bit better, being in centres that are running trials. So there is some data on that. There's closer monitoring, which for some people is good, for some people is a pain. And the helping future patients. So we've only got to where we've got to because of people who've been in trials before. I try, when I'm talking to an individual, that I don't think should come into it. So I try not to put a moral pressure on people going, well, you should, you know. So I usually never, no, actually I'd never mention that when I'm talking to people about trials. Uncertainty of outcome. So some patients just, you know what, you're talking about a trial. I don't even know if I'm going to get the experimental treatment. I don't know if it'll work. That uncertainty is just not for me. And that's fair enough, right? Some people don't want to do that. Unknown potential side effects, yeah. We know more about our standard treatments than we do about our trial treatments. Some extra visits. Some people find that reassuring. Some people, it interferes more with their life. A little less flexibility. We need to follow a protocol as best we can. And sometimes it's further to travel. So should you take part? I've made that seesaw deliberately, yes, you should. But that's because that's what I do. But for each person and with each trial, that's not necessarily the right thing. And often patients will ask me, there's a trial in so-and-so, is it worth me going there? And that's a conversation you need to have with your oncologist. So I'll stop there. I hope I've covered what a clinical trial is, talked a bit about the phases of clinical trials, life journey of a clinical trial, and then some various points to consider about trials. that all right? I'll stop there.